In this video, we are going to be looking at glycogen synthesis, and we're going to look at the control mechanism of glycogen cycling. So, when it comes to glycogen synthesis, understand that the term that is utilized to define it is glycogenesis. And as you can see, that is in the figure in red letters. So, understand that formation of glycogen is not the exact reversal of glycogen breakdown to glucose. What we have here is a series of steps that are going to be overall ending in the formation of UDP glucose, which is going to be the base, the unit that we utilize in order to synthesize glycogen. So when we have a meal that has increased amount of glucose, what's going to happen is that glucose is going to undergo the first step in glycolysis, which means that glucose is going to be converted to glucose 6-phosphate utilizing the enzyme hexokinase. Remember that this step of the reaction requires energy because we are going to be hydrolyzing an ATP molecule to ADP. Once we have glucose 6-phosphate, as you can see, phosphoglucomutase is actually going to be converting that glucose 6-phosphate into glucose 1-phosphate. Then, once we have the molecule of glucose 1-phosphate, then we are going to have the enzyme glucose 1-phosphate uridyl transferase going to transfer from UTP a UDP molecule onto glucose. Understand that the energy for the synthesis of glycogen, from glucose is provided by the hydrolysis of this UTP molecule. Glycogen synthase, even though it is the main enzyme that is utilized for synthesizing glycogen, understand that it actually requires a primer in order to do this synthesis. We have seen primers previously. In primers, we have seen them when we have DNA replication. Remember that there's an RNA primer that gets incorporated into the leading strand or lagging strand in order to have DNA polymerase polymerize the newly synthesized strand of DNA. Well, here our primer is going to look a little bit different. What we are going to have is that we're going to have a protein called glycogenin that is specifically at residue 194. The OH group in our tyrosine is going to be bonded to the anomeric position in UDP glucose. What this is going to happen is now form an acetal bond between glucose and the hydroxyl and the tyrosine at residue 194. Okay, so this means now that we're going to form this bond right here at the anomeric position in our glucose molecule. As you can see, UDP is going to be our leaving group here. So if we think about it, here the OH in the tyrosine is going to be our nucleophile. The carbon in the anomeric position is going to be our electrophile. And the UDP is going to be our leaving group. Understand that now that this glucose molecule has been incorporated into the hydroxyl group of tyrosine 194, which is here. Now, at position 4, if I number the carbon atoms that are around the glucose ring, we have carbon 1, 2, 3, 4, 5, and 6. Carbon 1 in the glucose that was incorporated into the glycogen can now react with a glucose or UDP glucose molecule that then it can repeat itself and load glucose molecules, onto this primer, Understand that this is going to be occurring until we have about 8 glucose molecules bonded together, then glycogen synthase is going to take over. So when glycogen synthase is going to be catalyzing the incorporation of glucose in specifically 1,4, okay, glycosidic bonds, understand that one of the things that happens is that in your UDP glucose molecules, the UDP is going to leave because that is a good leaving group. We are going to be forming an oxonium ion intermediate, and the carbon is going to be our electrophile. And in this instance, the hydroxyl group at carbon 4 is going to be our nucleophile. And that's how you make an alpha 1,4 bonds. But remember, glycogen molecule is actually a molecule that has branches. So on step 4, we are going to be forming those branches. So we have already discussed that the tyrosine, specifically in glycogenin, is going to be incorporating some glucose molecules utilizing as a substrate UDP glucose. Remember, after there is some incorporation of these glucose molecules, then glycogen synthase is going to take over. And as you can see, glycogen synthase also utilizes UDP glucose as a substrate to make specifically alpha 1,4 bonds. So when it comes to the formation of branches, understand that now what we are going to be utilizing are branching enzymes. And these branching enzymes, what they are going to be doing specifically is that they are going to be taking, so if we look at this residue in which we have a core strand of glucose molecules, they're all bonded together by alpha 1,4 bonds. The branching enzyme is going to take some of those residues and specifically make an alpha 1,6 bond. Then understand that further elongation at specifically the non-reducing ends are going to be catalyzed by glycogen synthase, making more alpha 1,4 bonds, and then the branching enzymes is going to continue to make branches at an alpha 6 bond, and that's how we build glycogen. Now, there are a list of glycogen storage diseases. And specifically, I want you guys to focus on type 1, type 2, and type 5. I am specifically going to talk about type 1. But I want you guys to do is to understand and practice if there's a defect in a particular enzyme that has to do with glycogen storage, why is it that we observe the effect in the glycogen in the organ, and also you can explain some of the clinical features that we observe. So I'm going to specifically talk about type 1. And as you can see, when it comes to type 1 is going to have a glucose 6-phosphatase or transport system deficiency. Remember, if we look at the pathway for specifically glycogen utilization, glucose 6-phosphatase is going to be preventing, if we have a defect there, is going to be preventing the release of glucose. Because remember, if we think about what is the role of glucose 6-phosphatase, this is the enzyme that exists in the ER and takes glucose 6-phosphate to glucose, then that free glucose gets transported outside the ER and it ends up in the bloodstream. So when we have type 1 of glycogen storage diseases, that I should specify, these are not common, but they are observed. in the type 1, our glucose 6 phosphatase is not going to be working. So one of the things that we observe is that when it comes to the glycogen in those affected organs, that in type 1, the specific things that get affected are liver and kidney, we're going to have an increased amount of glycogen, okay? As you can see, the clinical features of it is enlargement of liver, and I'm specifically going to talk about why is it that those patients have, for example, hypoglycemia and hyperlipemia. So if we rationalize what is occurring, I just established that in type 1, we have a glucose 6 phosphatase that is defective, which means that now we cannot release glucose, okay, in its free form during this process. Now, as you can see, this leads, as we can see in the table, to increased amount of glycogen. If we think about it, because we are keeping the levels of glucose 6 phosphate, and the phosphate I'm just going to write it as a P. Understand that even though some of it will be funneled into glycolysis, the other one is going to be funneled into glycogen synthesis, because we already explained that if you have excess glucose, then glucose 6 phosphate is going to be converted to glucose 1 phosphate, and that is going to end up forming UDP glucose, and that leads to the formation of glycogen. So that's why we have an increased amount of glycogen. Now, why is it that even though we have increased amounts of glycogens, this person are hypoglycemic? And remember, hypoglycemia means that this person is going to have abnormal low glucose levels. Now, if we think about it, remember that if we have a defect in the glucose 6 phosphatase, now your glucose cannot be released into the bloodstream to be processed through glycolysis. And that's why we have low glucose levels, because again, the phosphatase is not able to dephosphorylate your glucose 6 phosphate. So it leads to making more glycogen, and you don't have a lot of release of glucose, just the glucose molecule, into the bloodstream. Now, when it comes to hyperlipemia, this specifically is when you have an excess of triglycerides and cholesterol. So the interesting thing here is that, remember, if we have a surplus of glucose 6 phosphate, then some of it we already explained that ends up making glycogen, okay? The remainder of it is going to be converted into pyruvate through glycolysis. But understand that even though we're going to go later into the process of lipid metabolism and lipid synthesis, one of the things that occurs is that these biomolecules can be diverted into the conversion of triglycerol or triglycerides and cholesterol. So that's why overall in type 1 glycogen storage diseases, because our glucose 6 phosphatase is not working efficiently, we are going to have not a lot of glucose going to be released into the bloodstream. That's why the hyperglycemia. Some of it gets converted into glycogen because now we have an excess of glucose 6 phosphate. And understand that in the processing of this glucose 6 phosphate, if we have a lot of it, also we make triglycerides, and that can also lead to the formation of cholesterol, and that's why we have hyperlipemia. As I mentioned, please make sure that for the exam, you go through the same process of understanding how the defective enzymes, for example, type 2, which is going to be alpha-1,4 glucosidase, and phosphorylate, which is observing type 5, is going to be creating the effects that are outlined in this table. Now we are going to be discussing the allosteric regulation for glycogen synthase and phosphorylase. So overall, remember that when it comes to these enzymes, phosphorylase is going to be involved in the breakdown of glycogen, while glycogen synthase is going to be involved in the synthesis of glycogen. Now, as you can see here, we have things that are going to be activating these pathways, but we also have things that are going to be inhibiting these pathways. And they're going to be done in an allosteric regulation form. So, as you can see, glucose 6-phosphate, ATP, and AMP are going to be regulating glycogen phosphorylase and glycogen synthase. AMP specifically is going to stimulate phosphorylase to break down glycogen. But if we think about it, AMP means that the cell needs ATP. And because once we break down glucose, we make glucose 1-phosphate that then can be converted into glucose 6, and this can undergo metabolism in the form of respiration, that's how we are going to replenish that energy that is needed, that it was signaled by our AMP. Okay? Glucose 6-phosphate, understand that will be plentiful when glucose itself is abundant. So it makes sense that if we have a lot of glucose, then that is going to stimulate glycogen synthase to make glycogen. Okay? Now, understand that ATP and the presence of glucose 6 is going to inhibit phosphorylase, because if you have plenty of those molecules, the cell doesn't need to make free glucose to be processed to respiration and then producing more energy. Now, understand that there's other signals that we need to be familiar with that stimulate glycogen breakdown. And those are going to be epinephrine and glucagon. These two molecules are going to be signaling the breakdown of glycogen. So here on the right side, we have the structure of epinephrine and glucagon. As you can see, the difference between the two is that epinephrine is going to be specifically a molecule, okay? And then glucagon, it is specifically a peptide, so it's peptide base. As you can see, glucagon is going to be stimulating the liver, specifically glycogen breakdown, when the blood glucose is specifically low. Epinephrine, on the other hand, is going to be enhancing the glycogen breakdown in muscle and liver to produce fuel for muscle contraction. Okay? And you guys don't need to know the specifics of how it's done that. I just illustrated it on the right side, but you can relate it to the diagrams that we discussed previously. The chemistry is not any different from what we saw before. Another way in which glycogen breakdown can be regulated is through regulatory cascades. Again, these regulatory cascades are related to what the molecules that we observed before. Here we have epinephrine and glucagon specifically being bonded to a receptor. So what's going to happen in the takeaway message from here is that, as you can see, glycogen breakdown through the process of a regulatory cascade, which means that these small molecules that we talked about before, epinephrine and glucagon, what they are going to be initializing is a series of phosphorylation reactions. And this series of phosphorylation reactions, as you can see, is going to lead to the activation of phosphorylase. So when we have a regulatory cascade, as you can see, that is regulated through phosphorylation reactions.